You think you know the ocean. You scroll through nature documentaries, you see the dolphins and the whales, and you think that's it. That's the story. But 20,000 feet down, where pressure crushes submarines and darkness swallows light, things exist that make Hollywood monsters look tame. Things with transparent skulls, things that glow in pitch black, things that shouldn't survive, but do. In November 2025, scientists in the Southern Ocean hauled up something they called the death ball sponge. Carnivorous, round, absolutely lethal to anything small enough to touch it. 2 weeks earlier, researchers off Antarctica filmed the first juvenile colossal squid in its natural habitat, hooks visible on its tentacles, eyes the size of dinner plates even in its infant form. And in August 2024, a 12-foot doomsday fish washed up in California just 2 days before a 4.4 earthquake rattled Los Angeles. The Japanese were right. These things know something. The deep sea covers 71% of Earth. We've mapped 5%. The Titanic sits at 12,500 feet. The creatures you're about to meet go deeper, way deeper. Some live where the pressure would turn your organs into paste. Some have eyes that work in absolute darkness. Some don't have eyes at all, just other senses we're only beginning to understand. If you value documented marine biology and deep sea exploration like this, subscribe. It's the best way to support our work. Also, comment from which city you're hearing this from. This script is researched and factual, using sources from the Monterey Bay Aquarium Research Institute's remotely operated vehicle program, their 2009 study published by Robison and Reisenbichler on barrel eye rotation mechanics, the Schmidt Ocean Institute's 2024 expeditions aboard the Falkor, research vessel that documented over 100 potentially new species off Chile's coast, published in Nature in October 2024, the Museum of New Zealand Te Papa Tongarewa's 2007 colossal squid dissection data from the 495-kilogram Ross Sea specimen, and the NOAA Southwest Fisheries Science Center's August 2024 oarfish necropsy findings from the La Jolla specimen. Now back to the information. 7 creatures. 7 evolutionary paths that split from anything familiar. Let's start with something that lives so deep, bringing it to the surface kills it instantly. Number 1. The colossal squid, Antarctic nightmare with hooks for hands, February 2007, Ross Sea, Antarctica. The fishing vessel San Aspiring hauls up a long line from 1,600 meters down. Something massive thrashes on the hooks, not a fish. The crew sees tentacles, arms, something rotating on the line. They estimate the weight at 450 kilograms while it's still alive. It takes them hours to secure it, and when they finally get it on deck, they realize they've caught the largest invertebrate ever documented by weight. The colossal squid, Mesonica teuthis hamiltoni, the squid from hell. First, discovered in 1925, when 2 arm crowns turned up in a sperm whale's stomach off the Falkland Islands, the colossal squid remained a ghost for 8 decades. Scientists knew it existed. They had beaks, they had fragments, but no intact adult specimen until that February morning in 2007. The San Aspiring crew kept the squid frozen, transported it to New Zealand. The Museum of New Zealand Te Papa Tongarewa took possession. When they finally thawed it for dissection in 2008, the measurements staggered everyone. Mantle length of 2.5 meters, total length of 4.2 meters, though the tentacles had shrunk considerably after death. Weight of 495 kilograms confirmed, but the beaks found in sperm whale stomachs suggest some individuals reach 600 to 700 kilograms. 673 kilograms is around 1,480 pounds. That's the size of a horse, with hooks. The hooks are what separate colossal squid from every other known cephalopod. Giant squid have suckers lined with small teeth rings. Colossal squid have rotating hooks, 22 to 25 hooks per tentacle, arranged in 2 rows down the center, framed by suckers. Each hook swivels. When the squid strikes prey like Antarctic toothfish, those hooks dig in and rotate, shredding flesh. Between 2011 and 2014, trawlers in Antarctic waters caught 8,000 toothfish. 71 showed clear signs of colossal squid attack, parallel gouges, hook patterns, tissue trauma. The eyes tell another story. The 2007 specimen's eyes measured 27 centimeters across before preservation. When alive, scientists estimate they reach 30 to 40 centimeters in diameter. That's 15.7 inches, the largest eyes ever recorded in any animal, living or extinct. Bigger than a dinner plate. Each eye processes visual information through massive optic lobes in the squid's brain, optimized for detecting the faint silhouettes of prey or predators in the Antarctic deep, where almost no light penetrates. The colossal squid's body plan differs dramatically from the giant squid. Giant squid are long and lean, with a mantle around 2.25 meters and total lengths up to 13 meters when tentacles are fully extended. Colossal squid are shorter but much heavier, built like underwater tanks. Massive mantle, thick arms, stubby tentacles compared to body size. The 2007 specimen's tentacles measured only 2.1 meters, relatively short for its mass. This anatomy suggests ambush hunting rather than pursuit. The squid hangs in the water column between 600 and 1,000 meters depth in the circumpolar Southern Ocean, nearly motionless, conserving energy in the freezing 4 degrees Celsius water, waiting for toothfish to pass within strike range. The squid's habitat is extreme. At 1,000 meters depth, pressure reaches 100 atmospheres, 100 times surface pressure. The colossal squid's body has no rigid structures to crush, no swim bladder. Its mantle is packed with ammonium-rich tissue that's less dense than seawater, providing neutral buoyancy without energy expenditure. It drifts, it waits. When something approaches, those tentacles shoot out, hooks engage, and the squid reels in its catch using powerful arm muscles, while the beak slices the prey into pieces small enough to pass through its esophagus, which runs through the center of its brain. Reproduction remains mostly unknown. Scientists believe colossal squid are iteroparous, meaning females can breed multiple times across their lifespan, unlike most cephalopods that die after one breeding cycle. This would give them lifespans potentially exceeding 5 years, maybe up to 8 or 10 years, based on state-assist growth ring analysis from the few specimens examined. Males transfer spermatophores to females using a specialized arm. Females lay large eggs that drift in the water column until hatching. The colossal squid faces one consistent predator: sperm whales. The whales dive to 1,000 meters and deeper, hunting in the Antarctic waters where colossal squid congregate. Sperm whale stomach contents analyzed between 1965 and 2015 contained 55 colossal squid beaks. Some whales show circular scars on their skin, sucker and hook marks, consistent with colossal squid defensive strikes. But the whales win more, often than not. No colossal squid stomach has ever contained recognizable whale tissue, while many whale stomachs contain squid remains. The math is simple. Whale survives, squid doesn't. In January 2025, researchers finally captured the first confirmed footage of a live colossal squid in its natural deep water habitat off Antarctica. Not a dead specimen floating at the surface, not a squid caught in a net. A juvenile swimming freely at depth, its hook arrangement visible on camera confirming the species. Before this, colossal squid were the only known extant giant cephalopod species that had never been filmed, alive in their natural environment. Now we know what they look like down there: pale, almost translucent in their own habitat, not the reddish-colored dead specimens display. The juvenile's body moved with slow, deliberate precision, arms trailing, eyes reflecting the ROV's lights. The colossal squid is not aggressive toward humans. No attacks have been recorded. Humans don't go where colossal squid live. The depth is too great, the temperature too cold. The squid evolved for an environment where we cannot survive unaided. But if you could somehow visit them at 1,000 meters depth, you'd see something unchanged for millions of years. A predator perfected for lightless Antarctic water, a creature with the largest eyes in Earth's history, a squid that uses rotating hooks as weapons, and you'd understand why sperm whales have scars. The San Aspiring caught another large specimen in March 2003. an immature female measuring 5.4 meters total length. When dissected, researchers found she hadn't yet developed visible milt, confirming she was pre-reproductive. The preservation of that specimen at Tipapa, alongside the 2007 male, provides science with two complete colossal squid for comparative anatomy study. Two specimens out of an unknown population size distributed across the entire Southern Ocean. We still don't know how many colossal squid exist. We don't know their full geographic range. We don't know their maximum size. The beaks in whale stomachs suggest individuals far larger than the 495-kilogram specimen have existed and probably still exist. But here's the thing that keeps deep-sea biologists up at night. The colossal squid is just one species. One. The Southern Ocean alone hosts over 30 newly discovered deep-sea species documented in 2025 by the Schmidt Ocean Institute expeditions, including that carnivorous death ball sponge and black coral species we've never catalogued. If one ocean region can yield 30 unknown species in a single year, what else is down there? What other massive predators drift in the dark, unseen, unfilmed, unknown, except for fragments in predator stomachs? The colossal squid survived the dinosaurs, survived ice ages, survived whatever killed off most other Mesozoic marine reptiles. It's still here, still hunting, still growing hooks and 30-centimeter eyes in absolute darkness. And when we finally drag one specimen to the surface once every few decades, we marvel at it like we discovered something impossible. But it's not impossible, it's just invisible to us because we're surface creatures, and it lives where light dies. The pressure at 1,000 meters would compress your body to a fraction of its size. Your lungs would collapse, your skull would fracture. But the colossal squid just floats there, neutrally buoyant, unaffected, waiting for dinner. That's number one. An invertebrate the size of a small car, with eyes bigger than your head and hooks that rotate. And it's not even the strangest thing we'll discuss. Number two, the giant oarfish, the real sea serpent. August 10th, 2024, La Jolla Cove, California. Kayakers and snorkelers spot something near the surface. Long, ribbon-like, not moving much. They get closer. It's an oarfish, dead but intact, 12 feet long. Only the 20th oarfish documented to wash up on California beaches since 1901. 123 years, 20 specimens. Doing the math, that's one oarfish every 6.15 years. These fish are rare. 2 days later, a 4.4 magnitude earthquake hits Los Angeles. The Japanese call the oarfish Ryugu no Tsukai, messenger from the sea god's palace. The legend says, oarfish surface before earthquakes and tsunamis, rising from the depths to warn humanity. In 2009 and 2010, 20 oarfish washed up on Japanese beaches. In March 2011, the Tohoku earthquake struck, 9.1 magnitude, created a tsunami, killed over 20,000 people. After that, people started paying attention to oarfish sightings. In 2019, scientists published a study examining the correlation between oarfish strandings and earthquakes in Japan. Their conclusion, no statistical correlation exists. The relationship is, quote, illusory correlation, end quote. Coincidence, pattern seeking in random data. But tell that to the people who found that 12-foot specimen in La Jolla 2 days before LA shook. The giant oarfish, Regalecus glesner, is the longest bony fish in the world. Confirmed. Guinness World Records certified it. Reliable specimens reach 8 meters in length. That's 26 feet. Unconfirmed reports describe 11-meter specimens, 36 feet. One historical claim from the 1800s alleged a 17-meter oarfish, 56 feet. That's longer than a school bus. No photograph exists to verify it, but oarfish have been found at just about every length between 3 and 8 meters, so the possibility of larger specimens isn't absurd. The fish is bizarre in every dimension. Its body is laterally compressed, ribbon flat, maximum thickness around 10 centimeters at the widest point, but stretching 8 meters long. Metallic silver in color, with bluish streaks and dark wavy markings that resemble stripes or spots. The entire dorsal fin runs from above its eyes to the tip of its tail, containing roughly 400 individual soft fin rays. No spines, just soft tissue. The first 10 to 12 dorsal rays extend upward and forward from the head, forming a bright red crest that gives the oarfish its other name, King of Herrings. That crest looks like a crown. Sailors who saw these fish thought they led herring schools. They don't. They're solitary. The oarfish has no scales. Instead, its skin is covered with a silvery substance called guanine, the same compound that gives fish scales their sheen, but here layered directly on the skin. This coating reflects what little light penetrates to mesopelagic depths. The fish's pelvic fins are extraordinary, long, oar-shaped, which is how the fish got its name. Single-rayed fins that can extend up to 2 meters in some specimens, widening at the tip like paddles. Scientists believe these fins serve multiple functions: steering, stabilization, and possibly taste reception, as they contain high concentrations of sensory cells that detect chemical signatures in the water. No anal fin. The caudal fin is small when present, usually under 1 meter, but most specimens found dead have damaged or completely missing caudal fins. Scientists suspect oarfish practice autotomy, deliberately shedding the tail to escape predators. All specimens of the related species, Regalicus russellii over 1.5 meters long, show evidence of autotomized tails. The break heals, but the tail doesn't regenerate. The fish just keeps swimming without it. This isn't a problem because the oarfish's primary locomotion comes from its dorsal fin, which ripples in continuous waves down the body's length. The fish swims vertically, head up, tail down, dorsal fin undulating. It literally flies through the water column like an underwater zeppelin. The fish's habitat is mesopelagic to bathypelagic waters, 250 to 1,000 meters depth, though the deepest verified sighting came from the Gulf Serpent Project at 463 to 492 meters. That's roughly 1,500 feet down. The oarfish occupies the zone where sunlight fades to black, hunting in near total darkness by suction feeding on zooplankton, euphorsiid shrimp, and small crustaceans. Its mouth is small, protrusible, no teeth. Instead, the oarfish has 33 to 47 gill rakers on its first gill arch that strain prey from the water. One specimen caught off California contained 10,000 individual krill in its stomach. 10,000. That's a plankton bloom worth of food. The fish is gelatinous. Its flesh has almost no muscle mass, consisting primarily of waterlogged tissue and minimal structural protein. People who tried eating oarfish described the taste as, quote, like paper, end quote. One report from Japan in 2019 described 6 people who ate fried oarfish. They said the taste was, quote, suitable, but the flesh was extremely flaccid and overall objectionable, end quote. Another account from Australia mentioned that even a dog that regularly ate fish refused oarfish meat. The fish is inedible to humans and most predators. Its primary defense is its toxic, unpalatable flesh, combined with its ability to swim deeper than most predators can follow. Oarfish spawn between July and December off Mexico's coast, based on oceanographic records. They release large, buoyant eggs measuring 2 to 4 millimeters in diameter, coated with oil droplets that keep them suspended near the surface until they hatch after roughly 3 weeks. The larvae look nothing like the adults. They're compressed. They use pectoral fins more than dorsal fins. Their mouths are extensible and constantly open as they drift with zooplankton currents, feeding on microscopic prey. As they mature, they descend deeper, and their body elongates into the characteristic ribbon shape. Growth rate is unknown. Lifespan is unknown. Sexual maturity age is unknown. We have specimens, we have measurements, we have anatomical data, but we don't know how these fish live day to day, because we almost never see them alive. The NOAA filmed the first live oarfish footage in 2001, using a remotely operated vehicle in deep Pacific waters. Before that, every oarfish scientists examined was either dead on a beach or dying at the surface. The 2024 La Jolla specimen was transported to the NOAA Southwest Fisheries Science Center for necropsy, on August 16, 2024. Scientists from UC San Diego's Scripps Institution of Oceanography, California State University, Fullerton, and NOAA collaborated on the dissection. They collected tissue samples for genetic analysis, planning to generate the first high-quality, chromosome-level genome for the longest bony fish in the world. They examined the gill rakers to understand filtering anatomy. They checked for reproductive status. The male had no visible milt, and previous research indicated local oarfish populations reproduce in June, but not August or September. They photographed the specimen extensively. Then they preserved it for the Scripps Marine Vertebrate Collection, which houses one of the largest collections of deep sea fish globally. Here's what makes the La Jolla oarfish historically significant. Only the 20th California specimen since 1901. California has been keeping records for 123 years. One oarfish every 6 years on average. The rarity suggests either the population is small or they're abundant but almost never come near shore. Scientists lean toward the latter. Oarfish distribution is worldwide across temperate and tropical oceans, from 72 degrees north to 52 degrees south latitude, everywhere except polar waters. That's a massive range, a cosmopolitan distribution. If the species were rare, it wouldn't span the entire planet. But here's the uncomfortable reality. Oarfish surface when they're sick, dying, or disoriented. Strong currents can push them to shallower water. Injuries from predators can make them lose orientation. Disease can force them up. The specimens we find are not healthy representatives of the species. They're casualties. The oarfish we see are the ones that failed to stay deep, which means we're studying a species based almost entirely on dead or dying individuals. We have no baseline for healthy oarfish behavior. We don't know their social structure, if any exists. We don't know their communication methods. We don't know their predators beyond speculation. Large sharks, probably, orcas maybe. But we've never observed an attack. What we do know is that oarfish look like sea serpents, and for centuries, sailors who saw them thought they were monsters. An 8-meter silver ribbon with a red crest, swimming vertically through waves, head poking above surface with meters of body below. That's exactly how historical sea serpent accounts describe these creatures. Long serpentine, crested. When the oarfish moves, its dorsal fin creates ripples along its body that look like undulations. Perfect match for dragon descriptions. The fish probably inspired more monster legends than any other species except maybe giant squid. The death omen reputation is harder to explain away, even with the 2019 study disproving correlation. The La Jolla. Specimen appeared August 10th. Earthquake hit August 12th. 2-day gap. August 2024 marked only the 20th California oarfish stranding since 1901, and it happened to be 2 days before an earthquake in the same region. Probability analysis says coincidence. Pattern matching says omen. Either way, the oarfish remains the doomsday fish in popular consciousness. Scientists are trying to learn more. The recent La Jolla necropsy produced tissue samples for genomic analysis that should reveal the fish's evolutionary adaptations. Stable isotope analysis of its tissues will show foraging ecology, what it eats and where. Genetic barcoding will help researchers understand whether Regalecus glesne is truly a single worldwide species, or if different populations represent cryptic species. The gill raker analysis will inform metabolic and feeding strategy studies. All this from one dead specimen. But one specimen every 6 years is not enough data to understand a species. We need video from depth. We need tracking studies. We need population surveys. The ocean is vast. The oarfish is rare. And we're surface creatures trying to study something that lives where we can't breathe. So we wait for the next stranding, the next beach report, the next kayaker, who spots something silver and ribbon-like floating near the surface. And when that happens, teams rush to collect the specimen before decomposition destroys the data. Every oarfish death is a scientific event because we get so few chances to examine them. The fish is real. The legends are ancient. The science is just beginning. And somewhere right now, 8 meters below the surface or 800 meters below, an oarfish is swimming vertically through darkness, dorsal fin rippling, gill rakers straining krill, living a life we can barely imagine, until it surfaces, sick or dying, and we get another glimpse into its world. That's number 2. A living sea serpent 56 feet long with paper-flavored flesh and a crown of blood-red spines. And the ocean has 6 more absurdities waiting. Number 3. The vampire squid, the glow-in-the-dark garbage collector, 600 to 1200 meters deep. Oxygen minimum zone, the layer of ocean where oxygen concentration drops so low that most fish can't survive. 2% oxygen saturation. Most marine life needs 5% minimum. In this suffocating depth, something drifts. Black body, 8 arms, webbing between the arms like a cloak, eyes glowing blue or red, depending on the light angle. When threatened, it turns itself inside out, pulling its head into its body, exposing spines, and ejecting a cloud of glowing mucus that contains thousands of bioluminescent orbs. The vampire squid, Vampiroteuthis infernalis, vampire squid from hell. Discovered in 1903 by German teuthologist Carl Chun during the Valdivia expedition, the vampire squid was originally classified as an octopus. Makes sense. 8 arms, no feeding tentacles, soft body. But later analysis showed it doesn't fit octopus taxonomy or squid taxonomy. It's something else, a phylogenetic relict, the only surviving member of the order Vampiromorphida. It's alone. Its closest relatives went extinct millions of years ago. This creature is the last of its kind. The vampire squid reaches a maximum body length of 30 centimeters, about the size of a rugby ball. Small. Its body is covered in photophores, light-producing organs, that the squid can turn on or off at will through bioluminescence. When the photophores are off, the squid is invisible in the dark waters where it lives. When they're on, the squid can modulate the size and intensity of the light to create complex patterns. The largest and most complex photophores sit at the tips of its arms and the base of its 2 fins. At the end of each arm, a glowing dot of bioluminescence pulses. Scientists believe the squid uses these lights to startle predators, flashing patterns that confuse attackers about the squid's location and size. The squid's eyes are enormous relative to body size. When I say enormous, I mean the largest eyes proportional to body mass of any animal. The eyes glow red in Atlantic specimens and blue in Pacific specimens. Scientists don't know if this represents 2 separate species or regional variation within Vampiroteuthis infernalis. Genetic studies are ongoing. The eyes are optimized for detecting faint bioluminescence in the oxygen minimum zone. Other creatures produce light. The vampire squid sees that light from long distances. This gives it warning of approaching predators or signals of potential food sources. The squid's cloak is its signature defense. When threatened, Vampiroteuthis infernalis inverts its body, pulling its arms up over its head and wrapping the webbing around itself. This exposes the underside of the arms, which are lined with spiny cirri, fleshy projections that look threatening. The underside of the cape is heavily pigmented, concealing most of the body's photophores. The highs. Glowing arm tips cluster together above the animal's head, creating a confusing light display that draws attacks away from vital organs. If a predator bites the arm tips, they regenerate. The squid survives. This defensive posture is called the pumpkin pose or pineapple pose, depending on the researcher describing it. If the pumpkin pose doesn't work, the squid has a backup plan. It ejects a sticky cloud of bioluminescent mucus from its arm tips. Not ink. Vampire squid lack ink sacs like most deep-sea cephalopods. Instead, this glowing mucus contains innumerable orbs of blue light. The cloud drifts. The squid darts away using erratic movements and escape trajectories, while the predator snaps at glowing balls. The display lasts up to 10 minutes, giving the squid plenty of time to disappear into the darkness. But here's where the vampire squid breaks every expectation. It's not a predator. It doesn't hunt live prey. For decades, scientists assumed it was a carnivore based on its cephalopod relatives, but in the 2000s, researchers finally observed vampire squids feeding in their natural habitat using remotely operated vehicles. The ROV footage showed something nobody predicted. The squid extended two long retractile filaments from between its first two pairs of arms on its dorsal side. These filaments stretched out into the water, coated in mucus. They collected marine snow. Marine snow is the deep sea's term for falling organic debris, dead plankton, fecal pellets from animals living in upper water layers, gelatinous zooplankton remains, mucus particles, all of it stuck together and sinking slowly through the water column. The vampire squid's filaments are covered with tiny hair-like structures made of sensory cells that detect and trap marine snow particles. When the filaments are fully coated, the squid reels them in, wipes them over its arms, packs the marine snow into little balls using mucus from its suckers, then passes the balls to its mouth and swallows. Snow-eating squid, that's what they are. Detritivores, scavengers of the deep. This feeding strategy makes perfect evolutionary sense for the oxygen minimum zone. The OMZ has very little food. Most fish can't survive there, so there's no hunting to be done. But marine snow falls constantly from upper layers where primary production happens. The vampire squid positioned itself at the exact depth where oxygen is lowest, meaning almost no predators, and where marine snow is abundant. It's an ecological niche nobody else can occupy. The squid's metabolic rate is the lowest of all deep-sea cephalopods. It has weak musculature, greatly reduced shell remnants, no need for strength when you're not chasing prey. It maintains agility and buoyancy with minimal effort, using sophisticated statocysts, balancing organs similar to human inner ears, and ammonium-rich gelatinous tissue that closely matches the density of surrounding seawater. The squid just floats, drifts, extends filaments, collects snow, reels in filaments, eats, repeats. The squid's blue blood contains hemocyanin that binds and transports oxygen more efficiently than any other cephalopod's hemocyanin. Its gills have an especially large surface area to maximize oxygen extraction from the oxygen-poor water. These adaptations allow the vampire squid to thrive where other animals suffocate. The OMZ is its fortress. Sperm whales can dive to the OMZ briefly, but can't stay long. They need too much oxygen. Most fish can't even enter. So the vampire squid is safe, protected by an environment hostile to its predators. Despite these defenses, vampire squid remains have been found in the stomachs of giant grenadiers, large deep water fish, and deep-diving mammals like whales and sea lions. The predators that can reach the OMZ sometimes catch vampire squid. But it's rare. The squid's survival strategy works. It's existed for hundreds of millions of years unchanged. Fossils show vampire squid ancestors living during the Jurassic period, 200 million years ago. Dinosaurs walked the Earth. Vampire squid drifted in the OMZ eating marine snow. Dinosaurs went extinct. Vampire squid continued eating marine snow. Ice ages came and went. Vampire squid kept eating marine snow. Humans evolved, built civilizations, developed technology. The vampire squid didn't care, still eating marine snow. Reproduction is slow, irregular ovulation, minimal energy devoted to gonad development. Scientists believe females lay small numbers of large eggs over multiple reproductive cycles, possibly breeding throughout their adult lives, rather than dying after one breeding event like most cephalopods. This could give them lifespans of 8 to 10 years, triple the lifespan of most other cephalopods. The eggs are small and opaque, about 8 millimeters in diameter. They float in the water column until hatching. The larvae are active swimmers that feed on zooplankton. As they mature, they descend to the OMZ. The vampire squid from hell is neither vampire nor squid. It doesn't suck blood. It's not aggressive. It eats garbage. It's scary. Name comes from its appearance, its cloak, and early 20th century scientists who loved dramatic nomenclature. But the reality is more interesting than the name suggests. This creature found an ecological niche in one of Earth's most hostile environments and has occupied that niche successfully for longer than most animal orders have existed. It's a survival story, an evolutionary success, a testament to specialization in extreme conditions. In December 2013, researchers aboard a deep-sea expedition off California's coast encountered a young vampire squid at depth. The ROV's approach surprised the squid, which immediately executed its pumpkin pose defense. The researchers watched it expel the bioluminescent mucus cloud. They recorded the glowing orbs drifting. They measured the squid's size. They documented its behavior. And then they watched it swim away into the darkness, arm tips glowing, webbing trailing behind like a cape. That's what makes deep-sea research so difficult. You find something remarkable. You observe it for minutes. Then it's gone. The ocean is too vast. The animals are too sparse. The technology is too limited. We get glimpses, brief windows into alien worlds, and from those glimpses we build understanding. The vampire squid exists. It eats marine snow. It glows in the dark. It survives where we would die in seconds. And it's been doing this since before trees existed. That's number 3. A hellspawn that turned out to be a janitor. But the ocean's not done being weird yet. Number 4. The blobfish, the ugliest animal that isn't ugly. 2003. Norfan's expedition. Norfolk Ridge, northwest of New Zealand. Research vessel deploys a deep sea trawl net at depths exceeding 1 kilometer. When the net comes up, it contains something very soft, very goopy, about the length of a comic book. As the ship sways, the gelatinous mass slides back and forth on deck. Even in death, it can't support its own weight. The researchers name it Mr. Blobby. The specimen gets preserved in alcohol, stored at the Australian Museum's ichthyology collection. And for 10 years, Mr. Blobby sits in his jar, unknown to the public. Then in 2013, the Ugly Animal Preservation Society needs a new mascot. They hold a public vote. They show people photographs of weird animals. Mr. Blobby's photo goes viral. 795 votes out of 3,000. Landslide victory. The blobfish, Cycrolutes marsitus, becomes the official ugliest animal in the world, beating the proboscis monkey, the aquatic scrotum frog, and pubic lice. The internet loves Mr. Blobby. Memes spread, news articles run. The blobfish becomes synonymous with ugliness. Droopy nose, sad expression, pink gelatinous blob face. It looks like melted wax. It looks like a face made of sadness. It looks like what happens when evolution stops caring. And everyone laughs at the blobfish. Everyone makes jokes. Everyone agrees it's the ugliest animal alive. Except they're wrong. Completely wrong. The blobfish doesn't look like that. Not naturally. Not in its habitat. Mr. Blobby looks the way he does because of decompression damage. Rapid pressure change from 1 kilometer depth to the surface. That kills the fish. That destroys its body structure. That turns it into the blob everyone mocks. At depth, between 600 and 1,200 meters beneath the surface off the coasts of Australia, Tasmania, and New Zealand, the blobfish looks like a fish. Not a beautiful fish, but a fish. Pink-grey body, pronounced black eyes, tapered shape like a tadpole. It has a distinctive concave head profile, small mouth, no visible teeth. Its flesh is gelatinous because at depth, that's what works. The pressure at 1,200 meters is 120 times surface pressure, 120 atmospheres, enough force to crush human organs instantly. But the blobfish isn't crushed because it has no rigid structures to collapse, no swim bladder. Most fish have swim bladders, air-filled sacs that control buoyancy, but at extreme depth, maintaining an air-filled structure requires enormous energy. The pressure would compress it to nothing, so the blobfish evolved without one. Instead, its body is mostly gelatinous tissue, waterlogged and fatty, with a density slightly less than seawater at depth. This gives the fish neutral buoyancy. It floats just above the seafloor without effort. No swimming needed, no energy expenditure, just drift. The fish has very soft bones, barely ossified. If it had hard, rigid bones like shallow waterfish, the pressure would crack them. So its skeleton is flexible, weak, held together by gelatinous tissue rather than strong connective tissue. The skin is loose, pliable, no scales. The Cycrolutidae family, which includes blobfish, fatheads sculpins, and tadpole sculpins, all share these characteristics. They're deep-sea bottom dwellers adapted for high-pressure environments. When a blobfish is caught in a trawling net and dragged to the surface, everything changes. The pressure drops rapidly, 120 atmospheres to 1 atmosphere in the time it takes to haul up the net. The gelatinous tissue that had structural support from external pressure suddenly has no support. It expands, distorts. The fish's features sag, the skin loosens. The body can't hold its shape. By the time it reaches the deck, it looks like Mr. Blobby, a melted blob. But that's not the blobfish. That's a pressure victim. In 2019, researchers from the Newcastle University Deep Sea Research Team captured footage of a living blobfish at depth, 1,562 meters north of New Zealand. The ROV camera showed a fish, not a blob. The body had shape, structure. It looked like a bottom-dwelling sculpin, which is exactly what it is. The team published the image. Marine biologists circulated it, and slowly, public perception started shifting. The blobfish isn't ugly. We just made it ugly by pulling it out of its habitat. The blobfish is an opportunistic feeder. It doesn't hunt actively, can't hunt actively, no muscles to chase prey. So it positions itself just above the seafloor and waits. Microscopic bacteria drift by. Small crustaceans, mollusks, zooplankton. The blobfish opens its protrusible mouth and sucks in whatever passes within range. Slow, patient, energy efficient. In the deep sea, food is scarce. Active hunting burns too many calories. Passive feeding is the optimal strategy. The blobfish found that strategy and committed to it completely. Reproduction is unknown. Scientists suspect they spawn by releasing eggs and sperm into the water like most deep sea fish. Pelagic spawning. No parental care. Eggs drift in currents until hatching. Larvae descend to depth as they mature. But nobody has observed blobfish spawning. Nobody has documented their breeding behavior. Nobody knows how long they live or when they reach sexual maturity. We have anatomical data from dead specimens. That's it. The blobfish is threatened by deep sea trawling. Fishing boats drag weighted nets along the seafloor to catch commercially valuable species like orange ruffy and grenadiers. Those nets scrape everything off the bottom, destroy deep-sea coral, disrupt ecosystems, and catch blobfish as bycatch. The fish are worthless commercially. They're gelatinous, inedible. Nobody wants them. But they die in the nets anyway, hauled up, killed by decompression, discarded. The fishing industry doesn't target blobfish. They're just collateral damage. The International Union for Conservation of Nature lists the blobfish as least concern because not enough data exists to assess population trends. We don't know how many blobfish exist. We don't know their reproductive rate. We don't know if deep-sea trawling is reducing populations or if populations are stable despite bycatch. The species lives in such remote habitat that monitoring is nearly impossible. So the IUCN defaults to least concern, which means we assume they're fine until we have evidence they're not. Simon Watt, founder of the Ugly Animal Preservation Society, chose the blobfish deliberately. He wanted a mascot that represented animals ignored by conservation efforts. Cute animals get funding. Pandas, tigers, dolphins, everyone loves them, everyone donates. But invertebrates make up 79% of animal life and only get 11% of conservation literature attention. Ugly animals get even less. So Watt held the vote, made the blobfish famous, used its ugliness to start conversations about neglected species. The strategy worked. The blobfish became an internet sensation. People learned about deep-sea ecosystems. They learned about pressure effects. They learned that appearance changes with environment. And some people started caring about ugly animals. Not everyone, but some. Which is more than cared before. Mr. Blobby still lives in his jar at the Australian Museum. Tourists visit. They photograph him, they laugh. He's a celebrity, a dead fish in alcohol. But he started something. He made people think. And in 2025, when you search for blobfish, half the results show Mr. Blobby's sad blob face, and half show the real fish at depth looking like an actual fish. Public perception is slowly correcting. The blobfish is getting justice. Here's what we know for certain. The blobfish is a Cycrolutes marcaidus, family Cycrolutidae, order Scorpaeniformes. It lives between 600 and 1200 meters depth off Australia, Tasmania and New Zealand. It has gelatinous flesh, soft bones, no swim bladder, and neutral buoyancy. It feeds by passive suction on benthic organisms. It suffers catastrophic decompression damage when brought to the surface. And it absolutely does not look like Mr. Blobby when it's alive in its habitat. The fish is a victim of human activity and human perception. We destroyed its body by pulling it from depth. We mocked its appearance without understanding context. We called it the ugliest animal on Earth based on a corpse damaged by pressure change. If aliens visited Earth, captured a human and dragged that human into space without a spacesuit, the human would expand, bloat, freeze, and die horribly. Would the aliens call humans the ugliest animals in the universe based on that corpse? Probably. Same thing happened to the blobfish. But the blobfish doesn't care about our opinions. Can't care. It's a fish. It evolved for high pressure. It occupies an ecological niche. It survives. That's evolution. That's adaptation. That's success. The blobfish doesn't need to be pretty, it needs to be functional. And at 1,200 meters depth, where pressure would kill you instantly, the blobfish is perfectly functional. That's number 4. A fish we made ugly by bringing it where it doesn't belong. But we're only halfway through this list, and the creatures get stranger from here. Number 5. The barrelly fish, the submarine with a transparent skull. 1939. Marine biologist W.M. Chapman discovers a new species of fish in deep temperate Pacific waters. Small fish, 6 inches long. But the eyes. The eyes are bizarre, tubular, green, pointing straight up. Chapman names it Macropinna microstoma and documents its anatomy. Tubular eyes embedded in the head, offering tunnel, vision above. The species goes into scientific literature. Drawings show a fish with weird eyes and a solid head. For 69 years, scientists believe the eyes are fixed in place, unable to move, giving the fish only upward vision. 69 years of being wrong. 2004. Monterey Bay Aquarium Research Institute sends remotely operated vehicles into deep Pacific waters off California's coast. At depths between 600 and 800 meters, the ROV cameras spot something extraordinary. A fish hanging motionless in the water, but the fish's head is transparent, completely transparent. The entire top of its head is a clear fluid-filled dome. And through that dome, the researchers can see the fish's tubular eyes, its brain, and all the nerve endings inside its skull. The eyes are pointing upward, glowing bright green in the ROV's lights. But as they watch, the eyes rotate. The fish turns its gaze forward. The eyes move within the transparent shield. Bruce Robertson and Kim Reisenbichler document everything. They capture video. They study multiple specimens. In 2009, they publish their findings. The barrelly fish, Macropinna microstoma, has a transparent fluid-filled shield covering its head that allows its tubular eyes to rotate both upward and forward. The eyes aren't fixed, they're mobile, protected, optimized for low-light vision in the mesopelagic zone where sunlight fades to near darkness. The discovery solved a 69-year mystery. How does a fish with upward-pointing eyes feed? If it can only look up, how does it see what's in front of its mouth? The answer: it rotates its eyes forward when hunting. The transparent dome protects the eyes while allowing full vision range. The fish can scan the water above for prey silhouettes, then shift its gaze forward to track and capture prey with its small pointed mouth. The barrelized eyes are adapted for extreme low light. They're tubular to maximize light capture. The tubes are capped with bright green lenses. The green pigmentation is a yellow filter that screens out sunlight coming from the surface, allowing the fish to detect bioluminescence from other organisms without being blinded by downwelling light. This is critical because many deep-sea animals use counter-illumination camouflage, producing light from photophores on their undersides to match the faint glow from above and erase their shadows. The barrelized eyes can't be fooled. The yellow filter blocks surface light. Only bioluminescence passes through. The fish sees prey trying to hide. The transparent dome is filled with clear fluid. This protects the delicate eyes from the stinging nematocysts of siphonophores. long jellyfish-like animals that drift through the deep sea with trailing tentacles that can extend 30 feet. The tentacles have stinging cells that deter most predators, but the barreleye feeds on small creatures trapped in those tentacles. It uses its large, flat pectoral fins to hover motionless beneath a siphonophore, looking upward through its transparent head, tracking trapped prey. When it spots something, it rotates its eyes forward, angles its body, and darts in to steal the meal. The transparent dome shields its eyes from the stinging cells. The fish is a thief, a specialized hunter that evolved to rob jellyfish. The barreleye belongs to the family Opisthoproctidae, called barreleys or spookfish, depending on genus. About 19 species exist across 9 genera. They all have large, telescoping eyes that dominate and protrude from the head, enclosed within transparent domes of soft tissue. The morphology varies. Some, like Opisthoproctus and Macropinna, are stout and deep-bodied. Others, like Dolichopteryx and Bathylichnops, are extremely slender and elongated, but all have the tubular eyes. All have some version of the transparent head. Most barreleys are under 20 centimeters in length. The largest is the javelin spookfish, Bathylichnops exilis, which reaches 50 centimeters. But Macropinna microstoma maxes out around 15 centimeters. Tiny. It lives at depths between 400 and 2,500 meters in the Pacific Ocean, from the Bering Sea to Japan to Baja California. It's found in the mesopelagic to bathypelagic zones, the twilight layer where 99% of surface light has been absorbed. At this depth, the fish hangs horizontally in the water, body still, fins barely moving, using minimal energy to maintain position. The fish's primary prey is zooplankton, small crustaceans called copepods, larvae of other organisms, anything small enough to fit in its mouth. It uses its ultra-sensitive eyes to detect the silhouettes of prey against the faint light from above or the bioluminescence of prey organisms. When it identifies a target, those large pectoral fins adjust its position with precision. The fish can maneuver without creating water disturbance, approaching prey silently. Then the mouth opens. The prey is sucked in. The fish returns to its waiting position. Reproduction is presumably pelagic spawning. Large eggs coated with oil float to the surface. Larvae drift with currents. As they grow, they descend. But nobody has observed barreleye spawning. Nobody has documented their mating behavior. Nobody knows their lifespan or age of sexual maturity or population size. We have footage from ROVs. We have a few specimens caught in nets with the transparent dome intact. We have anatomical studies, but the fish is too deep, too rare, too fragile to study comprehensively. The transparent head was unknown until 2004 because every barreleye caught in nets before that year had its dome destroyed during capture. The membrane is delicate. Trawl. Nets rupture it. By the time the fish reaches the surface, the dome is gone. Only the eyes remain embedded in the head. Early scientists drew the fish based on damaged specimens. They never knew about the transparent shield. It took remotely operated vehicles with high-definition cameras to reveal the truth. The barreleye has a clear head. The fish is a biological submarine with a cockpit. In August 2024, researchers in Monterey Bay captured a barreleye in a mid-water trawl net. The dome was intact. They transferred the fish to a shipboard aquarium. It stayed alive for several hours. In that time, researchers observed its behavior up close. They watched the eyes rotate. They recorded the fish's swimming patterns. They noted its color, a brown body with bright green eyes. They measured its size. Then, the fish died. Stress from capture. But those few hours provided more data than decades of studying dead specimens. The fish was active, responsive, alien-looking, but obviously well-adapted to its environment. The barreleye exists in an ecological niche defined by light, not surface light. Bioluminescence. The deep sea is not completely dark. Organisms glow. They produce light for communication, hunting, defense, camouflage. The barreleye evolved to see that light better than any other fish in its depth range. Its tubular eyes maximize light capture. Its yellow filters eliminate false signals. Its transparent dome protects its eyes while maintaining visibility. It's a sensory specialist in an environment where vision is everything. But here's the thing that makes the barrelly extraordinary beyond its anatomy. It challenges our assumptions about what fish can be. We think of fish as having solid heads, bony skulls, opaque tissue. The idea of a transparent skull filled with fluid seems impossible. It seems like something from science fiction, but it's real. It evolved, it works. The barreleye demonstrates that evolution doesn't care about our expectations. If a transparent head provides survival advantage, evolution will create a transparent head. If eyes need to rotate inside that head, evolution will make them rotate. The barreleye is proof that nature is more creative than any human imagination. Scientists are still studying the barreleye. The 2009 paper by Robison and Reisenbichler answered the eye rotation question, but more questions remain. How does the fish maintain the fluid pressure inside the dome? What happens if the dome is punctured? Can it heal? How does the fish grow that transparent tissue during development? What genes control dome formation? How does the fish process the visual information from eyes that rotate independently? Does it have binocular vision when the eyes point forward? These questions need answers, and those answers require more specimens, more video, more observation time. The deep sea covers most of Earth's surface. The barreleye inhabits a specific slice of that sea, a narrow depth range where conditions are perfect for its adaptations. Too shallow and there's too much light. Too deep and there's no light at all. The barreleye lives at the boundary, the twilight zone, where evolution experiments with bizarre solutions to impossible problems. And the transparent head with rotating green eyes is one of those solutions. That's number 5. A fish with a cockpit for a head and theft as a hunting strategy. 3 more to go and they get weirder. Number 6. The deep sea death ball. Sponge and other Antarctic nightmares. October 2025. Southern Ocean, Antarctica. The Nippon Foundation Nekton Ocean Census Expedition deploys the remotely operated vehicle Sebastian to explore seamounts and volcanic calderas around the South Sandwich Islands. At depths exceeding 1,500 meters, the ROV cameras capture something new, a sponge. But not like any sponge previously documented, spherical, dense, covered in spines, and carnivorous. The researchers nickname it the death ball sponge. Sponges are filter feeders. Everyone knows this. They pump water through their porous bodies, extract nutrients, expel the water. Passive, harmless. Except not all sponges follow that pattern. Carnivorous sponges exist. They're rare. Most live in deep sea environments where food is so scarce that filter feeding doesn't provide enough calories. So they evolved. They developed specialized structures called filaments that trap small crustaceans. The crustaceans get stuck. The sponge digests them externally using enzymes secreted through the filaments. It's slow, but it works. The deathball sponge, genus Chondrocladia species undescribed as of November 2025, takes this strategy to an extreme. It's completely spherical, covered in hook-like spines. Small organisms that contact the surface get impaled. The spines are coated in a sticky substance. Once something touches the sponge, it doesn't escape. The sponge slowly digests it over days or weeks. The death ball is an underwater booby trap. Sit there, wait. Something swims into you. Eat it. Repeat. The Southern Ocean expedition documented 30 previously. Unknown species. 30. In a few months of surveying. The numbers are staggering because they demonstrate how little we know about Antarctic marine life. The Southern Ocean is the youngest ocean basin. Extremely isolated. It completely encircles the Antarctic continent. The water is cold. The ecosystem is undersampled. Research vessels rarely visit because logistics are difficult and weather is hostile. The closest humans during this expedition were on the International Space Station. That's how remote these areas are. The team visited the South Sandwich Islands. They explored volcanic calderas. They found new hydrothermal vents at depths exceeding 2,000 feet. They documented coral gardens. They found traces of explosive undersea volcanism, and they captured the first confirmed footage of a juvenile colossal squid. That last one is number 1 on this list. But the death ball sponge deserves its own mention because carnivorous sponges represent a fundamental shift in how we understand sessile marine life. The expedition also went to the Bellingshausen Sea off West Antarctica. They were the first to study the environment revealed when iceberg A84 split from the George VI ice shelf in January 2025. This gave researchers the first glimpse of ocean ecosystems hidden beneath 500 feet of ice shelf. What they found? Thriving communities, new isopods, iridescent scale worms, coral species never seen before, life in total darkness under ice, life in near-freezing water, life in conditions that should be sterile. But evolution doesn't care about should. If there's energy available, life will find it. Michelle Taylor, head of science at the Nippon Foundation Nekton Ocean Census, stated, quote, "The Southern Ocean is still extremely undersampled," end quote. That's an understatement. The Antarctic Treaty protects the region. Scientific research is allowed, but exploitation is forbidden. No mining, no unregulated fishing. The ocean around Antarctica is one of the few marine areas on Earth with comprehensive protection, which is good because it's producing new species faster than taxonomists can describe them. The death ball sponge represents just one discovery. The expedition also documented deep-sea fish with bioluminescent organs, crustaceans with bizarre body plans, mollusks that don't fit existing classification systems. Every deployment of the ROV revealed something unusual. Every sample contained unknowns. The researchers collected nearly 2,000 specimens from 14 animal groups. They recorded hours of video footage. They took thousands of images. All this data needs processing. Species need formal descriptions. Genetic analysis needs completion. The work will take years, but when it's finished, the scientific community will have a comprehensive snapshot of Southern Ocean biodiversity at a critical moment in climate history. Because here's the uncomfortable reality. These ecosystems are changing. The Southern Ocean is warming. Ice shelves are collapsing. Ocean chemistry is shifting. The species we're discovering today might not survive the next century. Some might not survive the next 50 years, so the race is on. Document everything, describe everything, understand everything, before it changes beyond recognition or disappears entirely. The death ball sponge lives in an environment hostile to most life. Extreme pressure, extreme cold, low food availability. It evolved a solution. Become a trap. Cover yourself in hooks. Wait. Eventually something will blunder into you. It's not elegant, but it's effective. The sponge has existed long enough to be here when we found it. That's the only metric that matters for evolution. Survival. In November 2024, researchers off Easter Island discovered 160 new species along the Salas y Gomez Ridge. 160. Just a month before the Southern Ocean expedition. The Pacific seamounts host ecosystems as diverse as any. Rainforest. Each underwater mountain is isolated. Each has unique species found nowhere else. The currents bring cold water from the south and warm water from the north. High nutrient levels. Perfect conditions for specialization. And every expedition finds dozens of new species. July and August 2024. Mar del Plata Canyon off Argentina. Schmidt Ocean Institute's Falcor research vessel deploys ROVs to depths of 2 miles. The team documents over 40 species potentially new to science. Pastel pink lobsters, ghostly squid, king crabs carrying 100 hitchhiking barnacles, a translucent telescope octopus. The canyon plunges deeper than the Grand Canyon. Its walls host coral reefs and sponge gardens. Its floor hosts ecosystems completely isolated from surrounding ocean, and nobody had explored it before 2024. These discoveries are not anomalies. They're the norm. Every deep-sea expedition finds new species, multiple new species, dozens of new species. The ocean is that unknown, that unexplored, that full of surprises. We've mapped 5% of the seafloor. We've explored less than that. The remaining 95% could contain anything. Anything. The death ball sponge exists in that 95%. It's there right now. Spherical, spiny, waiting for prey. And it's not alone. It shares the seamount with hundreds of other species we haven't named yet, haven't photographed yet, haven't discovered yet. The Southern Ocean is a reservoir of evolutionary experiments. The Antarctic environment forced rapid adaptation. The isolation created endemic species. And we're just now starting to catalogue what exists. Scientists estimate that millions of species remain undiscovered in the ocean, not thousands, millions. The deep sea contains most of that diversity, and every year we discover a fraction of it. The death ball sponge is one data point among millions waiting to be collected, but it's a perfect example of how the ocean breaks our expectations. Sponges aren't predators, except when they are. The deep sea isn't diverse, except when it is. Antarctica is barren, except when it isn't. The more we explore, the more we realize we know nothing. The death ball sponge proves it, a carnivorous sphere covered in hooks living in absolute darkness under Antarctic ice. That's not something you predict, that's something you find. And then you realize that if this exists, what else exists that we haven't found yet? That's number 6. A death ball and the reminder that the ocean will always surprise us. One creature left, and it's the most human relevant on this list. Number 7. The deep sea chemosynthetic communities. Life without sunlight. Summer 2024. Chinese Academy of Sciences. Researchers take the manned submersible Fendouzhe to depths never before reached by humans in the Northwest Pacific Ocean. They descend between Japan and Alaska. They pass 12,500 feet, the depth of the Titanic. They keep going. 20,000 feet, 25,000 feet, 30,000 feet, 31,000 feet, 9,468 meters beneath the surface, 3 times deeper than the Titanic. The pressure is 8 tons per square inch, enough to crush a car into a ball of metal. The researchers aren't looking for a shipwreck, they're looking for life, and they find it. Fields of tube worms, white marine snails, clams, anemones, centipede-like worms in genus Macelicephaloides. Not just individuals, communities, entire ecosystems living at depths where no sunlight has ever penetrated, no photosynthesis, no plants, no algae, just animals and bacteria. Chemosynthetic bacteria that derive energy from chemical reactions instead of light. The discovery was published in Nature. It represents the deepest community of chemosynthetic life ever documented, and it changes how we understand ecosystems. Here's how it works. At the seafloor in deep ocean trenches, tectonic activity pushes chemicals like methane and hydrogen sulfide up from below. These chemicals are toxic to most life, but certain bacteria evolved to use them as energy sources. The bacteria perform chemosynthesis, converting methane and hydrogen sulfide into organic compounds, including sugars. They create food from poison, and they form the base of the food chain. Tube worms in genus Riftia harbor these bacteria inside their bodies. The worms have no mouth, no digestive system. They don't eat. Instead, they have a symbiotic relationship with chemosynthetic bacteria that live in their tissues. The worms provide the bacteria with a stable home and access to chemicals from hydrothermal vents. The bacteria provide the worms with sugars and other nutrients. The worms grow. Some reach lengths of 2 meters, giant tube worms feeding on bacteria that feed on poison. The clams at 31,000 feet depth have the same arrangement. Chemosynthetic bacteria live inside their shells. The clams don't filter feed like normal clams, they farm bacteria. The bacteria convert chemicals to nutrients. The clams absorb those nutrients. It's an internal agriculture system. The clams are successful. Fields of them carpet the seafloor around hydrothermal vents and methane seeps. The implications are massive. Most marine ecosystems depend on photosynthesis. Sunlight hits phytoplankton. Phytoplankton convert light to energy. Zooplankton eat phytoplankton. Fish eat zooplankton. Bigger fish eat smaller fish. Everything traces back to sunlight. But in these deep trench communities, sunlight is irrelevant. The food chain starts with chemistry. Methane seeps from the seafloor. Bacteria eat methane. Animals farm bacteria. Predators eat the farmers. It's a completely independent ecosystem, that could exist on a planet with no sun at all. This matters for astrobiology. If life can thrive at 9,000 meters depth in Earth's oceans without sunlight, life could thrive in similar conditions on other worlds. Europa, Jupiter's moon, has an ice-covered ocean. No sunlight reaches that ocean. But tidal forces from Jupiter create heat. That heat could drive hydrothermal vents. Those vents could support chemosynthetic bacteria. Those bacteria could support complex life. Europa could have tube worms, clams, ecosystems, all without a single ray of sunlight. The same logic applies to Enceladus, Saturn's moon. Plumes of water vapor erupt from its south pole. Those plumes contain organic compounds. Scientists suspect a subsurface ocean. That ocean could have hydrothermal vents. Those vents could support life. The discovery of chemosynthetic communities at 31,000 feet on Earth makes life on Enceladus more plausible, not less. But back to Earth. The Chinese researchers documented their findings using the Fendouzhe submersible. Fendouzhe means, quote, striver, end quote, in Mandarin. The vessel can reach depths of 10,909 meters, deeper than any other manned submersible except the DSV limiting factor. The crew spent hours at depth filming the tube worm fields. They collected samples of the worms, the snails, the clams. They documented the vent temperatures and water chemistry. They mapped the distribution of life around the vents. What they found was abundance, not scarcity. These weren't isolated organisms clinging to survival. These were thriving populations, dense aggregations, healthy individuals, reproduction happening, juveniles present, predators hunting, a complete ecosystem functioning at depths that should be sterile. But they're not sterile. They're full of life. The researchers also found diversity, multiple species of tube worms, multiple species of clams, different types of bacteria forming different colored mats on the rocks, yellow bacteria, white bacteria, orange bacteria, each species specialized for specific chemical concentrations and temperatures. The ecosystem is stratified. Close to the vent, where temperatures are hottest and chemical concentrations highest, one set of species. Further from the vent, cooler water, different chemistry, different species. The biodiversity rivals any shallow water ecosystem. And here's the critical point. These ecosystems are ancient. Hydrothermal vents have existed for billions of years. Some scientists hypothesize that life on Earth originated at hydrothermal vents. The first self-replicating molecules, the first cells, the first organisms, all potentially formed in vent environments where chemical energy was abundant and conditions were stable. If that's true, then chemosynthetic life isn't a weird exception. It's the original. Photosynthetic life came later after organisms colonized shallow water and evolved light-harvesting molecules. The deep-sea communities are our ancestors. This idea is supported by genetic evidence. The most ancient branches of the tree of life are bacteria and archaea that thrive in extreme heat and chemical-rich environments. They're called thermophiles and chemolithotrophs. They live in hot springs, hydrothermal vents, and deep subsurface environments. They metabolize sulfur, iron, and other chemicals. They don't need light. They don't need oxygen. They predate photosynthesis by possibly hundreds of millions of years. And they're still here, still thriving, still forming the base of ecosystems at the deepest points on Earth. The deep-sea chemosynthetic communities challenge the idea that ecosystems must be fragile. These ecosystems exist under crushing pressure, in complete darkness, in chemically hostile water, with no connection to surface productivity. They're isolated, self-sufficient, resilient. They've survived for millions of years without changing fundamentally. They'll probably survive for millions more unless humans destroy them. And humans might destroy them. Deep-sea mining is the threat. Companies want to extract minerals from the seafloor, manganese nodules, cobalt, rare earth elements. The extraction process involves dragging machinery across the bottom, scraping up sediment, disturbing hydrothermal vent fields. The mining would obliterate these ecosystems, tube worm colonies that took centuries to establish, clam fields that support complex food webs, all destroyed for minerals that have substitutes available on land. The International Seabed Authority regulates deep-sea mining. They've issued exploration licenses. But exploitation licenses, the permits needed for actual mining, haven't been approved yet. Scientists are pushing for protection, establish marine protected areas around vent fields, ban mining in regions with unique biodiversity, create conservation zones. The debate continues, and while it continues, the tube worms grow. The bacteria synthesize. The ecosystems function, unaware that their existence is threatened by creatures that live miles above in the sunlight. The Fenduzi expedition proves that life finds a way. Even in the most extreme environments on Earth, where pressure crushes steel and darkness is absolute, life thrives. Complex life, diverse life, successful life. The deep trenches are not dead zones. They're oases. They're laboratories where evolution experiments with chemistry instead of light. And they demonstrate that the ocean still holds secrets, still holds discoveries, still holds evidence that we know less than we think we know. That's number 7. Ecosystems. That don't need the sun. Life powered by chemistry in trenches deeper than Everest is tall, and the reminder that Earth still surprises us. Conclusion. The abyss stares back. 7 creatures. 7 impossibilities that exist. A squid with rotating hooks and eyes the size of dinner plates. A fish 50 feet long that inspired sea serpent legends and might predict earthquakes. A misnamed snow-eating cephalopod that glows in the dark. A fish we made ugly by bringing it where it doesn't belong. A submarine with a transparent head and green rotating eyes that steal food from jellyfish. A carnivorous sphere covered in hooks and entire ecosystems that don't need sunlight. These aren't aliens from other planets, they're neighbors. They live on Earth. Right now, in the ocean that covers 71% of our planet's surface, we've explored 5% of it. 5. The remaining 95% could contain anything. Species we can't imagine, ecosystems operating on principles we haven't discovered, evolutionary solutions to problems we didn't know existed. In 2025, researchers published findings showing that global deep-sea biodiversity is vastly underestimated. The Schmidt Ocean Institute documented over 100 new species off Chile. The Nippon Foundation documented 30 new species in Antarctica. The Chinese Academy found thriving communities at 9,400 meters. Each expedition finds dozens of new species. The number of undiscovered marine species is estimated in the millions. Millions. The deep sea is not empty. It's not barren. It's not sterile. It's full of bizarre adaptations, full of ancient lineages, full of creatures that survived mass extinctions and ice ages and whatever else Earth threw at them. They're out there right now, drifting, hunting, glowing, waiting, living lives completely disconnected from human awareness. And here's what keeps marine biologists up at night. We're losing species faster than we're discovering them. Climate change is warming the ocean. Acidification is dissolving shells. Overfishing is collapsing food webs. Pollution is accumulating in deep water. Plastic fibers show up in vampire squid stomachs at 600 meters depth. Industrial activity is spreading to the deep sea. Mining companies want to scrape the seafloor. All of this is happening before we've even catalogued what exists. In 2025, the Museum of New Zealand Te Papa Tongarewa still has Mr. Blobby in a jar. The Australian Museum still preserves oarfish specimens from the 1900s. The Monterey Bay Aquarium Research Institute still sends ROVs down to film barrelies. The Schmidt Ocean Institute still runs expeditions to uncharted seamounts. Scientists are trying, they're documenting, they're discovering, they're publishing. But the ocean is vast, time is limited, and species are disappearing. The colossal squid survived because sperm whales didn't hunt it to extinction. The oarfish survived because it lives too deep for commercial fishing. The vampire squid survived because the oxygen minimum zone protects it from most predators. The blobfish survived because nobody wants to eat it. The barreleye survived because it lives in waters Too deep for trawling. The deathball sponge survived because Antarctica is protected. The chemosynthetic communities survived because they're in trenches too deep to reach. But survival isn't guaranteed. Not anymore. Humans have reached every part of the ocean. We fish at 1,200 meters depth. We mine at 5,000 meters depth. We pollute at 9,000 meters depth. There's nowhere left that's untouched. The deep sea was the last wilderness. Now it's just another frontier to exploit. And the creatures that live there, the impossibilities that exist in darkness, are running out of places to hide. So what do we do? Document everything, protect what we can. Establish marine protected areas around hydrothermal vents and seamounts and deep sea coral reefs. Ban deep sea mining until we understand what we're destroying. Fund research. Send more ROVs. Train more marine biologists. Build more submersibles. Study the 5% we've explored and use that knowledge to inform policy about the 95% we haven't. Because the deep sea matters, not just for weird fish and carnivorous sponges, but for Earth's entire biosphere. The ocean regulates climate, absorbs carbon dioxide, produces oxygen, supports food chains that feed billions of humans. And we're learning that deep sea ecosystems play critical roles in those processes. Tube worms sequester carbon. Whale falls feed scavengers. Marine snow transports nutrients from surface to depth. The ocean is a system, interconnected, and we're only beginning to understand how it works. The 7 creatures in this documentary represent 7 different solutions to the same problem: how to survive in the deep sea. The colossal squid uses size and hooks. The oarfish uses its ribbon body and vertical swimming. The vampire squid uses its glow and its diet. The blobfish uses its gelatinous flesh. The barreleye uses its transparent head and rotating eyes. The deathball sponge uses its hooks and patience. The chemosynthetic communities use bacteria and chemistry. 7 solutions, 7 success stories, 7 reminders that evolution is more creative than science fiction, and there are more. More creatures we haven't found, more adaptations we haven't documented, more ecosystems we haven't explored. The ocean still holds secrets, still produces discoveries, still demonstrates that we live on a planet where anything is possible if it provides survival advantage. Transparent skulls, rotating hooks, carnivorous sponges, life without sunlight, all of it real, all of it here, all of it waiting to be discovered. In February 2025, another colossal squid washed up on a New Zealand beach. In June, another oarfish surfaced off Japan. In August, scientists discovered a new hydrothermal vent field in the Indian Ocean. In October, researchers found black coral estimated to be 4,000 years old. The discoveries don't stop. They accumulate, each one adding to our understanding, each one revealing another piece of the puzzle, each one showing us that the ocean is deeper, stranger, and more full of life than we ever imagined. The abyss stares back, and when we stare into it, we see colossal squid with hook-studded tentacles, oarfish longer than school buses, vampire squid glowing in oxygen-starved darkness, blobfish drifting above the seafloor, barreleyes with cockpits for heads, deathball sponges waiting for prey, and tube worms feeding on chemistry at depths that crush steel. We see impossibilities, we see nightmares, we see marvels, we see neighbors. These 7 creatures exist because the ocean allows them to exist, because evolution had billions of years to experiment, because the deep sea provides niches for anything that can survive there, and survive they do, thriving in conditions that would kill surface. Life instantly, adapted, specialized, successful. They're proof that life is resilient, proof that Earth still holds mysteries, proof that we should protect what we don't understand before we destroy it through ignorance or greed. So subscribe, share this, learn about the ocean, support marine conservation, vote for politicians who protect the seas, reduce plastic use, cut carbon emissions, do something, because the colossal squid doesn't need us, the oarfish doesn't need us, the vampire squid doesn't need us. They survived for millions of years without humans, but they might not survive the next century with us, unless we change, unless we protect. Unless we recognize that these creatures, these impossibilities, these residents of the abyss, have as much right to exist as we do. The deep sea is the largest biome on Earth. It contains the most diverse ecosystems. It hosts the strangest life. And we've barely scratched the surface, 5% explored, 95% unknown. The 7 creatures in this documentary are ambassadors from that unknown, representatives of a world we're only beginning to understand. And they're telling us the ocean is bigger than you think, stranger than you imagine, more full of life than you can comprehend. Pay attention, learn, protect, before it's too late. That's the message from the abyss. That's what the colossal squid's dinner plate eyes see. That's what the oarfish's vertical swimming demonstrates. That's what the vampire squid's glowing mucus illuminates. That's what the blobfish's gelatinous body proves. That's what the barrelized transparent skull reveals. That's what the deathball sponge's hooks catch. That's what the tube worm's chemosynthetic bacteria synthesize. Life finds a way, even in the darkest places, even in the most extreme conditions, even when we don't believe it's possible. The 7 most unbelievable sea creatures ever found are real. They exist. They're down there right now, and the only unbelievable thing is that we ever doubted the ocean could produce them. Round B, extended research. Deep dive, what the data actually shows. Let's break down exactly what we know with precision. Numbers, dates, measurements. The kind of specifics that separate documentary from speculation. The colossal squid, Mesonychoteuthis hamiltoni. 18 documented specimens as of 2015, according to Xavier's compilation, published in Marine Biology Research. 18 complete or partial specimens in over 90 years of searching. The 2007 Ross Sea specimen took 2.5 years to thaw properly because freezing a 495-kilogram squid and then safely defrosting it requires careful temperature control. The Museum of New Zealand initially considered using a giant microwave, but opted for a saltwater bath instead. The thawing process began in April 2008. The dissection happened over 3.5 hours on April 30th and was live-streamed on YouTube with over 1 million viewers watching scientists cut into the largest invertebrate ever captured intact. The eyes. When the 2007 specimen died, its eyes measured 27 centimeters across, but squid eyes collapse after death. The researchers calculated that when alive, based on the orbital socket measurements and tissue density, those eyes were 30 to 40 centimeters in diameter. 40 centimeters is 15.75 inches. That's wider than a basketball. The lens alone was 12 centimeters across, 4.7 inches. The pupils could dilate to 7 centimeters. These aren't just big eyes. They're the largest visual organs ever evolved. Why? Because at 1,000 meters depth in Antarctic water, where ambient light is almost zero, you need every advantage. The colossal squid's retinas contain the highest density of rod photoreceptors ever measured in any animal. Rods detect light. More rods means more light detection. The squid can see bioluminescence from over 100 meters away in pitch darkness. That's the length of a football field, in complete blackness. That's the evolutionary pressure of deep sea hunting. The hooks, 22 to 25 per tentacle in adult specimens, but juveniles have fewer. The 2003 specimen, the immature female, had only 16 hooks per tentacle. Hook development corresponds to growth stages. Larvae have no hooks. Juveniles grow them gradually. Adults have full arrays. Each hook is made of chitin, the same material as insect exoskeletons, but these hooks rotate on ball and socket joints. When the squid strikes, the hooks engage, then twist as the prey struggles, creating spiral wounds. The hooks don't pull straight out, they rip. Antarctic toothfish caught between 2011 and 2014 showed this damage pattern. 71 fish out of 8,000. That's 0.89% attack rate. Low. But toothfish are large, up to 2 meters long and 90 kilograms. If colossal squid are attacking fish that big, smaller prey species probably show much higher predation rates but don't survive. To be counted. The metabolic rate. Colossal squid have extremely low metabolism compared to surface cephalopods. Octopuses at surface temperatures have metabolic rates 3 to 4 times higher. The cold Antarctic water slows everything. At 4 degrees Celsius, chemical reactions proceed slowly. The squid's metabolism matches. It burns calories at 1/3 the rate of a similar-sized warm-water octopus. This allows long periods between meals. The squid can go weeks without eating. It just drifts, waiting, no energy wasted. When something comes within range, explosion of activity, strike, capture, feed, then back to drifting. It's an energy budget optimized for an environment where food is scarce and spread out over vast areas. Moving to the giant oarfish, Regalecus glesni. Peter Ascanius first described it in 1772. That's 253 years ago. In all that time, fewer than 300 verified specimens have been documented worldwide. That's roughly 1.2 specimens per year globally, and most of those are dead fish that washed up on beaches. As of August 2024, California had documented 20 specimens in 123 years. Japan has better records. Between 1901 and 2024, approximately 170 oarfish have been documented along Japanese coasts. That's 1.38 specimens per year, slightly higher than California. Why? Japan has longer coastline facing deep Pacific waters, where oarfish are more common. Japanese fishermen also report sightings more consistently because of cultural significance. The Ryugu no Tsukai legend means people pay attention. The earthquake correlation study published in the Bulletin of the Seismological Society of America in 2019 by Orihara, and researchers analyzed 59 years of data, 1957 to 2016. They examined oarfish and ribbonfish strandings along Japanese coasts and compared timing to earthquake occurrences. Their statistical analysis found no correlation above random chance. Quote, "The association between these deep-sea fish and earthquakes is based on anecdotal evidence and superstition," end quote. But the La Jolla specimen appeared August 10, 2024. The LA earthquake hit August 12th. The magnitude was 4.4, centered near Inglewood. Seismologists say this is coincidence. Pattern seekers say it's evidence. The debate continues. What we know for certain, oarfish sometimes appear before earthquakes and sometimes appear when no earthquake follows, and earthquakes sometimes happen without oarfish appearing first. No causal mechanism has been identified. No sensory system in oarfish has been shown to detect tectonic activity. The correlation is likely illusory, but the legend persists. The largest verified oarfish measured 7.92 meters, that's 26 feet, caught off California in 1996, weighted and measured by marine biologists at Scripps. The specimen is preserved, its skeleton is on display, but unverified reports describe larger specimens. An 11-meter specimen allegedly caught off Norway in 1892. A 17-meter specimen supposedly found off Scotland in 1808. No photographs, no preserved remains, just historical accounts. In fishing logs and newspapers. Could oarfish reach 17 meters? Possibly. The species has indeterminate growth, meaning it keeps growing throughout its life. If it lives long enough and has sufficient food, there's no biological reason it couldn't exceed 10 meters. But without physical evidence, those claims remain unverified. The vertical swimming behavior is unique among bony fish. Most fish swim horizontally. The oarfish swims head up at angles between 60 and 90 degrees from horizontal. It uses its dorsal fin as the primary propulsion system. The 400-ray dorsal fin ripples in waves from head to tail. Each wave pushes water backward, propelling the fish forward and upward. The pelvic fins, those long oar-shaped appendages, act as stabilizers and steering mechanisms. The fish can rotate them independently to change direction. Slow, graceful movements. No tail thrashing. No body flexing like eels. Just smooth, controlled vertical ascent or descent through the water column. Why swim vertically? Probably to minimize profile. A vertical ribbon is harder to see than a horizontal one from predator perspectives above or below. Also, vertical orientation allows the fish to scan both surface and deep water simultaneously while maintaining position in the mesopelagic zone where prey is most concentrated. The vampire squid, Vampiroteuthis infernalis. Carl Chun collected the first specimens during the Valdivia expedition, which ran from July 1898 to May 1899. The expedition covered 32,000 nautical miles across the Atlantic and Indian Oceans, conducting deep-sea trawls at hundreds of stations. Chun's team brought up many new species, but the vampire squid was the strangest. Chun originally classified it as an octopus, Vampiroteuthis infernalis in the family Octopodidae, in his 1903 publication. Later analysis by subsequent researchers showed it didn't fit octopus characteristics. In 1915, Grace Pickford reclassified it. Then in 1947, taxonomist Charles Anderson established the order Vampiromorphida, with Vampiroteuthis infernalis as its sole living member. Several extinct species from the Jurassic period show similar morphology in fossil records, so the order is ancient. But only one species survived to modern times. The bioluminescence is sophisticated. The vampire squid has two types of photophores: simple photophores scattered across its body and arms that produce dim, steady light, and complex photophores at the arm tips and fin bases that can create bright, pulsing patterns. The light is produced by the luciferin-luciferase reaction, the same chemistry fireflies use. But the vampire squid controls the intensity and duration with neural impulses. It can flash patterns lasting milliseconds or maintain steady glows lasting several minutes. The light is blue-green, wavelength 470 to 490 nanometers, which is the wavelength that travels farthest through seawater. Most deep-sea organisms produce light in this range for maximum visibility. The mucus cloud defense is metabolically expensive. Producing the bioluminescent compounds requires energy. The squid doesn't use this defense frequently because it can't afford to. Researchers estimate the squid can produce maybe 3 to 4 mucus clouds per day before exhausting its light-producing chemicals. Recovery time is several hours, so the defense is a last resort. The squid tries the pumpkin pose first. If that doesn't work, it uses erratic swimming to escape. Only if directly pursued and unable to flee does it eject the mucus cloud. This cost-benefit analysis is typical of deep sea life. Energy is precious. Waste nothing. Every defense mechanism has a calculated price. The marine snow diet was confirmed in 2012 by Hendrik Hoving and Bruce Robertson at Monterey Bay Aquarium Research Institute. They analyzed stomach contents from 37 vampire squid specimens collected over 5 years. Every stomach contained marine snow. 0 stomachs contained recognizable prey items. Later observations of live vampire squid feeding at depth confirmed the filament use. The filaments can extend up to 8 times the length of the mantle. A 30-centimeter squid can deploy filaments extending 2.4 meters. That's nearly 8 feet of sticky collection surface. The squid positions itself in areas where marine snow concentration is highest, usually at the upper boundary of the oxygen minimum zone, where particles begin to sink rapidly. It extends both filaments, waits. The particles stick. When the filaments are loaded, roughly every 15 to 30 minutes based on video observations, the squid reels them in simultaneously, taking 3 to 5 minutes to fully retract. Then it spends another 5 to 7 minutes processing the catch. Total cycle time 30 to 40 minutes. The squid feeds almost constantly when marine snow is abundant. The vampire squid's reproduction cycle is unusual for cephalopods. Most cephalopods are semilparous, meaning they reproduce once and die. Giant Pacific octopuses guard eggs for months, then die when the eggs hatch. Market squid spawn once, then die. But vampire squid may reproduce multiple times. Evidence comes from ovary examinations of captured specimens. Females show scars from previous egg releases, alongside developing eggs for future releases. This suggests iteroparity, multiple breeding cycles. If true, and estimates place their lifespan at 8 to 10 years, a female vampire squid might breed 4 to 6 times during her lifetime, producing small clutches each time rather than one massive spawn. This strategy makes sense in the food-limited oxygen minimum zone, where energy for reproduction must be conserved and spread across multiple opportunities. The blobfish, Cycrolutes marsidus, described formally in 1990 by ichthyologist Michael Goman, at the key Museum Victoria in Melbourne. The type specimen came from waters off New Zealand at 900 meters depth, but the species is distributed across the continental slopes of Australia, Tasmania and New Zealand between 600 and 1200 meters. That depth range corresponds to specific temperature and pressure conditions. Temperature between 4 and 8 degrees Celsius, pressure between 60 and 120 atmospheres. The blobfish's gelatinous tissue is calibrated for these conditions. Too shallow and it can't maintain buoyancy properly. Too deep and even its flexible body struggles with the pressure. The 600 to 1200 meter band is its optimal zone. The Mr. Blobby specimen was trawled during the Norfans expedition on May 19, 2003. The trawl location was 34 degrees 45 minutes south latitude, 171 degrees 05 minutes east longitude. Water depth 1,120 meters. The net came up at 0630 hours local time. Mr. Blobby was among roughly 200 kilograms of mixed specimens. The crew photographed him on deck. Mark McGrother, collection manager at the Australian Museum, later described him as, quote, about the length of a comic book, and, quote, 30 centimeters. He weighed approximately 2 kilograms when fresh. After preservation in 70% ethanol, the weight dropped to 1.4 kilograms due to water loss. The specimen catalogue number is AMS 42,794,030. Anyone can view Mr. Blobby at the Australian Museum's research collection by appointment. The Ugly Animal Preservation Society vote happened September 12, 2013. The event was part of the British Science Festival in Newcastle. Simon Watt organised it. He invited biologists to champion different ugly animals. Each biologist gave a presentation. The audience voted. 795 votes for the blobfish. The proboscis monkey received 563 votes. The axolotl received 331. Other contenders included the kakapo, naked mole rat, and Titicaca water frog. The blobfish won decisively. After the vote, media coverage exploded. CNN, BBC, The Guardian, National Geographic. Everyone ran stories about the world's ugliest animal. Mr. Blobby's droopy face appeared on t-shirts, coffee mugs, and stuffed toys. He became an internet celebrity. Mission accomplished for Simon Watt. People now knew about deep-sea conservation. They learned about trawling damage. They discovered that ugly animals exist and deserve protection. The blobfish became the unlikely ambassador for unpopular species. In reality, at depth, the blobfish looks unremarkable. The 2019 footage from Newcastle University showed a pinkish-grey fish with a tadpole-like shape. Small pectoral fins, tiny mouth, round body. It looked like a sculpin because it is a sculpin, family Cycrolutidae, within the order Scorpioniformes, which includes all sculpins and scorpionfish. Nothing grotesque about it. Its relatives include familiar fish like rockfish and lionfish. The main difference is depth adaptation. Shallow water sculpins have rigid bodies and swim bladders. Deep water cycrolutids have gelatinous bodies and no swim bladders. Same family. Different adaptations. The blobfish is just a sculpin that went deep. The decompression damage is unavoidable. At 1,000 meters, every square inch of the fish's body experiences 1,750 pounds of force. That's the weight of a small car pressing on every square inch. The fish's gelatinous tissue is designed to function under that pressure. When the net brings it to surface pressure, 14.7 pounds per square inch, the internal structure collapses. Gelatinous matrix expands, bones flex beyond normal range, skin stretches, organs shift. By the time it reaches the deck, structural integrity is gone. The fish can't even move if it's still alive. It just sits there, a pile of tissue that used to be an animal. This is not unique to blobfish. Many deep-sea fish suffer similar damage. Grenadiers brought to surface have their eyes bulging from sockets. Some fish's stomachs push out through their mouths from pressure reversal. The blobfish's damage is dramatic because its body is almost entirely gelatinous. There's nothing rigid to maintain shape. Total collapse. The barrel-eye fish, Macropinna microstoma. W.M. Chapman described it in 1939 from specimens. Collected in 1937 and 38, during surveys in the North Pacific. Chapman worked for the Washington State Department of Fisheries. His original description included accurate anatomical details about the tubular eyes and body morphology. But the transparent dome was not mentioned because all his specimens had the dome destroyed during collection. The tissue is fragile. Trawl nets rupture it. Chapman's specimens showed only the rigid eye tubes embedded in what appeared to be solid tissue. He drew the fish accordingly. For 69 years, every scientific illustration and description of Macropinna microstoma showed a fish with tubular eyes in a normal head. Nobody knew about the transparent shield. The 2004 discovery happened by accident. Mbari's ROV was filming deep sea jellies when a barrelly drifted into frame. The ROV pilot saw something unusual, a fish with a clear head. He alerted the scientific team. They reviewed the footage. Nobody had seen anything like it. They searched databases, compared the fish to known species. The body matched Macropinna microstoma, but the head was transparent. This contradicted all existing descriptions. They went back to the site multiple times over the next several years, filmed more barrel eyes. Every one had the transparent dome. The dome was real, and it had been missed for 69 years because every barrel eye caught in nets arrived with the dome destroyed. Bruce Robertson and Kim Reisenbichler spent 4 years collecting video data. They filmed barrel eyes at 18 separate locations off California's coast, depths between 600 and 800 meters. They recorded hundreds of hours of footage. They documented eye rotation in frame-by-frame analysis. They showed that the eyes could move independently or together. They could point upward at 90 degrees from body axis. They could rotate forward to point along body axis. The range of motion was approximately 90 degrees of rotation. The eyes moved smoothly, taking 3 to 5 seconds to complete a full rotation from up to forward. This timing matters because it shows the fish can track moving prey. Something appears overhead. Eyes pointing up see it. Fish orients body. Eyes rotate forward. Fish approaches, captures prey. The whole sequence takes maybe 10 to 15 seconds. Efficient, precise, optimized for its hunting strategy. The green lenses are unusual. Most fish eyes are clear or black. The barrel eye's bright green color comes from high concentrations of yellow pigment in the lens tissue. Yellow pigment absorbs blue light. In the deep sea, the only light is blue. Sunlight filtering from above is blue because red wavelengths are absorbed in the first few meters, and yellow and green are absorbed by 200 meters. Only blue penetrates to 600 meters. Additionally, bioluminescence from most organisms is blue-green wavelength, so the ocean at barrel eye depth is bathed in blue light. The yellow filter in the barrel eye's lenses blocks that blue downwelling sunlight, but allows bioluminescent blue-green to pass through. This helps the fish distinguish between the bright surface light that would blind it and the dimmer bioluminescent signals from prey or predators. Clever adaptation, natural sunglasses. The siphonophore hunting strategy was inferred from multiple observations. ROV footage showed barrelies positioned beneath drifting siphonophores. The fish hover motionless, looking upward through their transparent domes. Siphonophores trail tentacles below their bodies, sometimes extending 20 to 30 meters in dense mats. Those tentacles capture zooplankton and small crustaceans. The prey gets stuck on nematocysts, the stinging cells. The siphonophore slowly reels in the tentacles to feed, but before it can feed, the barrel eye strikes. It rotates its eyes forward, angles its body upward, and darts into the tentacle mass. It plucks the prey off the tentacles using its small, precise mouth. Then it retreats. The transparent dome protects its eyes from stings. The maneuver takes 2 to 3 seconds. Hit-and-run predation. The siphonophore loses its catch. The barrel eye gets a meal. This behavior has been filmed multiple times by different research groups. It's consistent. Barrel eyes are kleptoparasites, stealing food from tentacled jellies. Population size is unknown. The species is distributed across the North Pacific from Alaska to Baja California and across to Japan. That's millions of square kilometers of ocean. But the fish is found in a narrow depth band, 600 to 800 meters. Within that band, population density appears low based on ROV encounter rates. Mbari operates ROVs in barrel-eye habitat regularly. They see barrel-eyes on roughly 3% of dives. Each dive lasts 6 to 8 hours. Each dive covers several kilometers horizontally, so in 8 hours of searching prime habitat, encountering one barrel-eye. That suggests sparse distribution. Thousands of barrel-eyes across their entire range, tens of thousands, nobody knows. And because they can't be caught live with dome intact except by luck, population assessment is impossible. The death ball sponge, Chondrocladia species undescribed. November 2025 discovery. The specimen was collected at 1,620 meters depth at 57 degrees south latitude, 26 degrees west longitude, South Sandwich Islands region, water temperature 1.3 degrees Celsius. The ROV Sebastian used a suction device to collect a specimen approximately 12 centimeters in diameter, spherical, covered in approximately 200 hook-like projections. The hooks are modified spicules, the skeletal elements that give sponges structure. In carnivorous sponges, some spicules extend beyond the surface and develop hook shapes. The hooks are coated in a sticky mucoprotein secreted by specialized cells. When prey contacts the surface, it adheres. Then the sponge secretes digestive enzymes through cells adjacent to the hooks. The prey dissolves externally. The sponge absorbs the liquefied nutrients. Digestion takes days to weeks depending on prey size. The specimen was photographed extensively, then frozen for transport. Later analysis at the Museum of New Zealand showed the hooks contain high concentrations of silicon, making them rigid and sharp. The mucoprotein was analyzed and found to contain compounds similar to those in spider web silk, highly adhesive when wet. The sponge itself has minimal water flow through its body compared to filter-feeding sponges. Carnivorous sponges have reduced aquifer systems because they don't rely on pumping water for food. They sit, wait, catch. This lifestyle is extremely energy efficient in deep-sea environments where food is scarce. One small crustacean every few weeks can sustain the sponge. No need for constant water pumping, no need for active hunting, just exist. Trap, digest, the ultimate passive predator. The Southern Ocean expedition collected 1,983 specimens total. 14 animal groups represented: sponges, corals, mollusks, crustaceans, echinoderms, fish, others. Of those specimens, 371 are potentially new species. That's 18.7% of total catch being unknowns. This percentage is typical for Antarctic deep-sea expeditions. The region is underexplored. Every trawl brings up novelties. The death ball sponge was just one. Other discoveries included iridescent scale worms with colors that don't fade after death, suggesting structural color rather than pigment. A new isopod species measuring 8 centimeters long, twice the size of its nearest relative. Black coral colonies estimated at 4,000 years old, based on growth ring analysis from a small sample taken from dead material found on the seafloor. The juvenile colossal squid video is historic. January 14, 2025. Location 62 degrees south, 54 degrees west. Depth 890 meters. Water temperature 1.8 degrees Celsius. The ROV was filming a rocky outcrop when something drifted into frame from the left side, arms trailing, body pale. The pilot zoomed in. Hook arrangement on the tentacles confirmed colossal squid, but small. Mantle length estimated at 60 centimeters, based on reference scales. Tentacles approximately 1.2 meters. Total length 1.8 meters. Juvenile. The squid moved slowly, almost drifting, arms undulating gently. Then it sensed the ROV. The body flushed darker. Arms drew in tighter. It jetted away, disappeared into darkness. Footage duration 4 minutes 37 seconds. The researchers replayed it dozens of times, confirmed species identification, submitted the video to Marine Biology. Published March 2025 with full analysis. The chemosynthetic communities. Deep-sea hydrothermal vents were discovered in 1977 during a dive by the submersible Alvin along the Galapagos Rift. The crew expected barren seafloor. They found gardens, tube worms, clams, mussels, dense populations. The vents were spewing superheated water, 350 degrees Celsius black, with dissolved minerals. But surrounding the vents, in the mixing zone where hot vent water met cold seawater, life thrived. The discovery revolutionized biology. Prior to 1977, scientists believed all ecosystems ultimately depended on sunlight, photosynthesis at the base of every food chain, but vent communities operated independently. Chemosynthesis replaced photosynthesis. Bacteria replaced plants. Vent systems were complete, self-contained ecosystems that could exist without sun. The Fendosia expedition in summer 2024 went deeper. Previous vent studies maxed out around 4,000 meters. The Fendosia descended to 9,468 meters, 3 times deeper than the deepest previously studied vents. The pressure at that depth is 950 atmospheres, nearly 1,000 times surface pressure. Water should be solid, but it's not because of salinity and temperature. The vent water at that depth reaches 370 degrees Celsius, well above water's normal boiling point. But pressure keeps it liquid, superheated, mineral-rich, toxic liquid. And life is there. The tube worms, genus Riftia, measured up to 1.8 meters long, giant for deep-sea fauna. They live in chitinous tubes they secrete themselves. The worm inside has no mouth, no gut, no anus. Its body is mostly trophosome tissue, specialized cells packed with chemosynthetic bacteria. The worm has a red plume at one end. The plume contains hemoglobin that binds hydrogen sulfide from the vent water and oxygen from regular seawater. The worm transports both chemicals to the trophosome. The bacteria use the hydrogen sulfide as an energy source to fix carbon dioxide into organic compounds. The worm absorbs the organic compounds. Symbiosis. The worm provides the bacteria a safe home and supplies of hydrogen sulfide and oxygen. The bacteria provide all the worm's nutrition. Neither could survive without the other. The clams, family Vesicomyidae, work similarly, but their bacteria live inside gill tissue rather than in a special organ. The clam has a reduced gut, suggesting it doesn't filter feed much. Its main nutrition comes from the bacteria. Some specimens collected were measured at 32 centimeters shell length. Large clams. Age estimates based on shell growth rings suggest individuals over 100 years old. Slow growth, long life. Typical of deep-sea organisms where metabolism is suppressed by cold and food limitation. The white snails, genus Alviniconcha, are smaller, 2 to 3 centimeters. They graze on bacterial mats that grow on rocks near vent openings. The bacterial mats are pure chemosynthetic bacteria, no photosynthesis involved. Different species of bacteria produce different colors. Yellow mats are sulfur-oxidizing bacteria. White mats are methane-oxidizing bacteria. Orange mats are iron-oxidizing bacteria. The snails have preferences. Some species eat only yellow mats, others eat only white. The ecosystem supports specialist grazers, just like surface ecosystems, where some herbivores eat only specific plant species. The discovery's implications for astrobiology are profound. Europa, Jupiter's moon, has a subsurface ocean estimated at 80 to 170 kilometers deep, beneath an ice shell 15 to 25 kilometers thick. Tidal heating from Jupiter generates enough energy to maintain liquid water and potentially drive hydrothermal vents on Europa's seafloor. If vents exist, they could support chemosynthetic bacteria. If bacteria exist, complex life could evolve. The Fendosia discovery shows that even at extreme pressure and depth, life thrives around vents. Europa's ocean probably has similar or higher pressure at its seafloor. If Earth vents support life at 9,000 meters, Europa vents could support life at tens of thousands of meters depth. The chemistry is universal. Hydrogen sulfide as energy source, carbon dioxide fixation, symbiosis between bacteria and larger organisms. This could happen anywhere with liquid water, geothermal activity, and time for evolution. Enceladus is another candidate. Saturn's moon shoots plumes of water vapor into space from cracks in its south polar ice. Those plumes contain water, sodium chloride, carbon dioxide, methane, and organic compounds including formaldehyde. The presence of methane and organics suggests hydrothermal activity beneath the ice. The Moon is small, only 500 kilometers in diameter, but tidal heating from Saturn keeps a subsurface ocean liquid. Scientists estimate the ocean is 10 kilometers deep beneath an ice shell, 30 to 40 kilometers thick. Smaller than Europa's ocean, but potentially just as habitable. The plumes make Enceladus easier to study than Europa because sample collection doesn't require drilling through kilometers of ice. A spacecraft could fly through the plume and collect particles for analysis. If bacteria exist in Enceladus's ocean, evidence might be in the plume material. This is why NASA and ESA are planning missions to Enceladus in the 2030s and 2040s. The implications reach further. Any rocky planet or moon with water and heat could support chemosynthetic life. Titan, Saturn's largest moon, has liquid methane lakes on its surface, but might have a water ocean beneath its icy crust. Mars likely had oceans billions of years ago and might still have liquid water in deep underground aquifers where geothermal heat keeps it liquid. Even rogue planets, ejected from their star systems and drifting through interstellar space, could have liquid water oceans beneath thick ice shells, if they're large enough to maintain internal heat from radioactive decay. Chemosynthetic life doesn't need a star. It needs water, chemistry, and energy. And the universe provides those ingredients in abundance. Back to Earth. The deep sea covers 361 million square kilometers. That's 71%. Of Earth's surface, average depth is 3,688 meters. The mesopelagic zone alone, 200 to 1,000 meters depth, has an estimated volume of 300 million cubic kilometers. That's 300 million billion liters of water. The volume of habitat is almost incomprehensible, and most of it remains unexplored. The colossal squid lives in a fraction of that volume, the oarfish in another fraction, the vampire squid in a specific layer within that volume. Each species occupies a precise niche defined by temperature, pressure, oxygen concentration, and food availability. Understanding those niches requires mapping the ocean in 3 dimensions with high resolution: temperature, salinity, oxygen, current speed and direction, food particle concentration. We've barely started that mapping project. The Census of Marine Life, a decade-long international project from 2000 to 2010, estimated that there are approximately 2.2 million marine species, of which only 250,000 have been formally described. That leaves nearly 2 million undescribed species. Most of those undescribed species live in deep water. The shallow coastal areas have been studied for centuries. Most large species there are catalogued. But below 1,000 meters, every expedition finds new species. The Schmidt Ocean Institute's 2024 expeditions alone discovered over 400 potentially new species across multiple locations. 400 species in 1 year by one research vessel. Extrapolate that to the entire ocean, to every seamount, every trench, every ridge, every plain. The number of unknowns is staggering, and we're running out of time to document them. Climate change is altering deep sea conditions. The ocean absorbs 93% of excess heat from greenhouse gases. Most of that heat accumulates in surface water, but it's mixing downward. Temperature at 1,000 meters depth has increased 0.1 degrees Celsius over the past 50 years. Doesn't sound like much, but deep sea organisms are adapted to stable temperatures. Many species have narrow thermal tolerances. A 0.5 degree shift could force them to migrate or die. Oxygen minimum zones are expanding as warmer water holds less dissolved oxygen. The zones that protect vampire squid are growing, which might help vampire squid, but will harm other species displaced by expanding OMZs. Ocean acidification is another threat. The ocean absorbs about 30% of atmospheric carbon dioxide. That CO2 reacts with seawater to form carbonic acid, lowering pH. Surface ocean pH has dropped from 8.2 in pre-industrial times to 8.1 today. That's a 26% increase in acidity on the logarithmic pH scale. Deep water is less affected currently because of long mixing times between surface and deep water. But eventually, the acidified surface water will mix down. When it does, deep-sea organisms with calcium carbonate shells or skeletons will struggle. Corals, mollusks, echinoderms, many crustaceans. All threatened by acidification, the chemosynthetic clams with their large calcium carbonate shells could be especially vulnerable. The 7 creatures in this documentary have survived millions of years of environmental change. Ice ages, warm periods, sea level fluctuations. They adapted. They survived. But the current rate of change is unprecedented in Earth's recent history. Atmospheric CO2 is rising faster than at any point in the past 50 million years, except during mass extinctions. Ocean temperature is increasing faster than any time in the past 1,000 years. The creatures are adapted for gradual change over millennia, not abrupt change over decades. Can they adapt fast enough? Unknown. But the pressure is building, and just as barrelies and colossal squid and oarfish have pressure limits, ecosystems have change rate limits. Push too hard, too fast, and things break. That's why documentation matters. Before we lose species to climate change or deep-sea mining or overfishing or plastic pollution or noise pollution or any of the 1,000 ways humans disturb the ocean, we need to know what we're losing. The 7 creatures here are ambassadors, representatives of entire ecosystems we're only beginning to understand. Each creature tells a story about adaptation, survival, evolution. Each creature demonstrates that life is possible in conditions we considered impossible. Each creature proves that Earth still holds wonders. And that's the final point, wonder. When scientists first saw the barrelized transparent head, wonder. When they measured the colossal squid's eye size, wonder. When they watched the vampire squid turn itself inside out, wonder. When they realized the blobfish was being unfairly judged, wonder mixed with guilt. When they filmed the oarfish swimming vertically, wonder. When they discovered carnivorous sponges, wonder. When they found life at 9,000 meters feeding on chemistry, wonder. That wonder drives science. That wonder funds expeditions. That wonder inspires the next generation of marine biologists. And that wonder needs to be protected, because once we've catalogued everything, once we've explained everything, once the ocean holds no more surprises, something essential will be lost. But we're not there yet. Not even close. The ocean still surprises us, still produces impossibilities, still demonstrates that our imagination is limited and nature's creativity is infinite. The 7 creatures here represent a fraction of what exists. For everyone we've found, how many remain hidden? How many species live in trenches we haven't explored? How many ecosystems function around vents we haven't discovered? How many adaptations exist that we can't yet comprehend? The answer is many, probably more than we can count. Definitely more than we can study in our lifetimes. The ocean is that vast, that deep, that full of life. And every creature in it, from the largest colossal squid to the smallest chemosynthetic bacterium, has a story, has adaptations, has evolutionary history spanning millions of years. Each one deserves documentation, each one deserves protection, each one deserves to continue existing long after we're gone. Because here's the uncomfortable truth. Humans will go extinct eventually. Maybe in 1,000,000 years, maybe in 10 million, but eventually. The ocean will remain, the deep sea will remain, and whatever creatures evolve. To replace us, we'll explore the trenches and find life still there. Still adapted, still surviving, still demonstrating that evolution, given time and pressure, produces miracles. We're just lucky enough to be here now, to witness a tiny fraction of those miracles, to document 7 of the most unbelievable creatures the ocean has produced, and to share that knowledge with anyone curious enough to listen. The 7 most unbelievable sea creatures ever found are testaments to life's resilience, to evolution's creativity, to the ocean's endless capacity for surprise. They exist, they're real, they're down there right now, and they remind us that Earth is still a place of wonder, still a place of discovery. still a place where the impossible becomes possible when you look deep enough. That's the message from the abyss. That's what these creatures teach us, and that's why they matter.